Today we're diving into a really fascinating piece of new research. It's about a simple sensor designed to detect one key hormone, and it could be a massive game changer for spotting a very dangerous type of pregnancy. Okay, let's get into it.
First things first, what are we actually talking about here? An ectopic pregnancy. So this is a situation where a fertilized egg implants somewhere it absolutely shouldn't, usually in a fallopian tube. And because it's not in the uterus where it's supposed to grow, this is an incredibly dangerous and potentially life-threatening condition.
And this isn't some rare one-in-a-million thing. Get this, in 2019 alone, there were an estimated 6.7 million new cases worldwide. That number, wow, that really puts the sheer scale of the problem into perspective, doesn't it?
So with millions of cases every year, you can see why early and accurate diagnosis is just absolutely critical. But how do you spot it? Well, researchers have been chasing down biological clues, and they've really started to zero in on one in particular. And that clue is a hormone called estradiol, which you'll often see shortened to just E2. Now, E2 is a major player in regulating the reproductive system. But what researchers have been discovering is that its levels can tell us a whole lot about the health of a pregnancy.
So what's the connection? Well, this quote from the study pretty much spells it out. They found that women with very low levels of estradiol, we're talking below 100 picograms per milliliter, were way more likely to be experiencing an ectopic pregnancy. The takeaway here is simple. Low E2 is a major, major red flag.
All right, so if measuring E2 is the key, why isn't this just a routine, simple test that everyone gets? Well, that brings us to what you could call the diagnostic bottleneck. It turns out getting a fast, accurate reading of this hormone is way harder than it sounds. And this slide just lays out the problem perfectly. On the one hand, you've got our current methods, which are slow, they're expensive, they need really complicated sample prep, and they often use some pretty nasty solvents. What we really need is everything on the right side of the screen. A test that's fast, affordable, simple to use, and safe. Something you could use right there at the point of care. And this is exactly where this new research comes into play. To try and solve this problem, a team of scientists developed a whole new kind of electrochemical sensor, one that was built from the ground up to be simple and super cost-effective. So what's the secret sauce? It's this molecule right here, a complex made from glutathione, which is a really common antioxidant, and copper. This little GCU complex is the heart of the new sensor, and it's what gives it the unique ability to detect estradiol.
And the best part? The process for making this stuff is just wonderfully simple. We're talking a one-pot recipe at room temperature. You literally just mix two solutions together, stir them for a few hours, filter out the blue solid that crashes out, and then let it dry. That's it. No fancy equipment, no extreme heat or pressure.
And here is the final product. This powder is that G-cup complex. You can even see its kind of rough, aggregated structure under the microscope here. This powder is then just mixed into a liquid and dropped onto an electrode to create the finished sensor. Super simple.
So how in the world does this actually work? Well, the mechanism is really clever, and honestly a little counterintuitive. See, it's not about the sensor producing a stronger signal, it's about seeing that signal get weaker.
The researchers call this suppression mode. Basically, instead of the estradiol causing a bigger electrical current, its presence actually suppresses it. It pushes the signal down. This graph shows it perfectly. See that red peak? That's the sensor's baseline signal. But now look at the blue peak. That's the signal when estradiol is in the mix. It's way lower, right? What's happening is the estradiol molecules are basically clogging up the sensor's surface and blocking the flow of electrons. So the more estradiol you have, the more blocking you get, and the lower the signal goes.
And the researchers confirmed this themselves right in their paper. They found this really clear relationship. As the amount of E2 goes up, the electrical current goes down. It's an inverse relationship, but it's a very, very reliable one.
So if we zoom all the way into the molecular level, what's going on? Well, this diagram gives us a clue. The estradiol molecule itself gives up an electron and a proton. That electron is then captured by the GQ complex on the sensor, and that whole interaction is what disrupts the electrical flow and causes that drop in the current we just saw. Okay, so the theory is super elegant, but does it actually work? This brings us to the final and, let's be honest, the most important part, the results, and what this all could mean for the future.
This sensor's sensitivity is frankly amazing. Its limit of detection, that's the smallest amount it can spot, is an incredibly low 0.125 picograms per milliliter in a clean buffer solution. But what's even more important is that even in artificial urine, which is a much messier, more complex mixture, it can still detect as little as 4.99 picograms per milliliter, and that is well within the range you'd need for a clinical diagnosis.
But can it pick out estradiol from a crowd of other molecules? This chart shows us its selectivity, and the answer is a big yes. When they tested it against other similar-looking molecules that might be floating around in a sample, like tyrosine and tryptophan, the sensor barely even reacted. It showed less than 10% interference, which means it is laser-focused on E2, and that is exactly what you need in a diagnostic tool.
So when you put it all together, you get a balance of incredible promise and some real-world challenges. On the pro side, the sensor material is cheap to make, it's super sensitive, and it's very selective. But the researchers are also upfront about the hurdles. They still need to figure out how to improve the long-term stability of the GCU complex and find ways to keep the electrode surface from getting gunked up or fouled after you use it a few times.
And all of this leaves us with one final big question. While there's still more work to do, this research presents a really powerful new approach. Think about it. A simple, affordable point-of-care test could empower doctors to detect these hormonal warning signs of an ectopic pregnancy way earlier than ever before, and that could potentially save lives. The question now is how quickly can we turn this promising new clue into a clinical reality?